to turn the stage, as it were, over to my colleague, Professor Ian Hill, a member of the Nobel Conference Committee, who is also going to introduce for us our next speaker. So I'm going to hand it over to Ian.
Pharmaceuticals and biologics play an integral role among a battery of treatments used in managing cancer, and the discovery of novel and effective drugs remains an active field of research and development in academic and corporate laboratories alike. Our next speaker, Dr. Jim Thomas, has built an extensive career in the biopharmaceutical industry and worked to see the discovery, development, and FDA approval of many biologic drugs. Following his PhD work at Purdue University and a postdoctoral fellowship in biochemistry, nutrition, and cell physiology at MIT, he began working as a scientist at Genentech. From there, he transferred to Immunex in Seattle to become the director of cell sciences and was there to see the FDA-approved Enbrel, a breakthrough monoclonal antibody biologic for the treatment of autoimmune diseases like rheumatoid arthritis, in addition to many other biopharmaceutical drugs. Dr. Jim Thomas ultimately became the Vice President of Process Science, which he continued through the buyout of Immunex by Amgen in 2002 and retiring as the Vice President of Process and Product Development. Since his retirement from Amgen, Dr. Thomas became a founding partner and CEO of Just-Evotec Biologics, which received $8 million in 2016 from the Bill and Melinda Gates Foundation towards its mission to expand access to life-changing protein therapeutics across the globe. He is now the Executive Vice President, Global Head of Biotherapeutics, and President of U.S. Operations of Just-Evotec Biologics. Ladies and gentlemen, Dr. Jim Thomas.
I want to thank the organizing committee for inviting me to speak at this Nobel Conference. It's quite an honor to be here. I'm going to provide some thoughts about how we're addressing the issue of global access to biologics today. And this is a rather complex issue, and I've tried to calibrate the talk to the average listener that's out there. I may be talking in an elementary way to some, but over the heads of others. And if you're in either of those camps, I apologize. But I hope I'm connected with the students that are out there, and I hope you see some opportunities to engage with your interest in continuing education to come in and make contributions to the field. This is a requisite forward-looking statement. The average cost to develop a novel therapeutic for the treatment of serious illness like cancer is well over $2 billion. So when this is translated to patients, the average cost could be $10,000 a month or more. And I think some of the newer therapies are actually quite a bit more than this. This is going to obviously create quite a hardship on patients that are out there in this country and other actually countries like Europe and Japan. I think we have to have very good insurance, but can be some significant co-pays and so forth associated with this. But if you think about across the planet, there are many billions, actually billions of people that won't be able to access these drugs at all. So we really need to do something about that. So to start, I want to provide some high-level view of why these drugs actually cost so much. At the very highest level, when you look at the biotech and biopharma industry as a whole, the process for discovering, developing, and commercializing new therapeutics is remarkably inefficient. And that's not because, there are a lot of inept people out there and they're not working very hard. That's not the case at all. The real reason is that the process is remarkably challenging and very complex. So any good scientist will tell you that there are far more failures in the lab than successes. Understanding the complexity of human biology and the fundamental mechanism of disease is quite a humbling experience, actually, and I can say that by personal experience. Now, the lead candidates that come out of discovery, only 10% make it past phase 2 clinical studies or the proof of concept stage. And for those drugs that make it to phase 1, only 5 to 20% of those drugs actually make it to the approval process. And unfortunately, cancer therapeutics are on the low end of that range. And it takes a long time, 8 to 12 years or more, actually, to go through this whole cycle. And then, obviously, to add to further complexity, these drugs are actually, therapeutics are going into people. And, they have to be safe, and we have to develop all the information preclinically and in the clinic to demonstrate that they're safe. That's very important, of course. And we work with the regulatory authorities to make that happen, FDA and other world regulatory authorities. So if you track the cumulative costs of developing all these new therapeutics, about 75% of the cost resides between discovery and clinical proof of concept studies, because that's where most of the failures happen. These are rather expensive experiments from discovery to proof of concept. There's a lot of those experiments that have to happen for a drug actually to move forward. And I think, the success rates have to be addressed. And there are a number of ways that we can do that. And I'm going to actually talk about some of those in some of the following slides. So what can we do about the high costs of therapeutics, particularly biotherapeutics or biologics? So this slide represents at a very high level this process from discovery to commercialization. We start at discovery, we improve the design of the molecule, we find methods and process methods for making these drugs. We do along the way, of course, we do a lot of pharmacokinetics and we do a lot of safety testing before we get to the clinic. Then we make the drug in clinical manufacturing, and we put that into people in these phase 1/phase 2 studies. Phase 1 is really for safety, to make sure that we're safe in people. And phase 2 is to try to get some hint of efficacy of the drug. Now, if the drug actually passes this stage, it goes to late stage clinical development, phase 3 clinical development, where we're refining the dosing. And we're also getting additional safety information before it's actually approved and becomes a commercial product. So better prediction of clinical efficacy at discovery is actually critical. So if you want to think about some ways of reducing the cost of these therapeutics, and there are a few ways I will talk about, it starts with getting better prediction. And that sort of makes sense, right? I mean, if you're doing discovery, you want to pick the molecule that's actually going to work. So we need better biology around that discovery process, and we're getting it. We're actually putting in a lot of robotics at that stage so we can generate data really efficiently, and we're actually using AI and machine learning tools, computational tools, to understand that biology better so that we can get better prediction. Another thing that we can do to increase our success and decrease our failure rate is the identification of biomarkers to segment patient populations. Now, biomarker is not the therapeutic target itself, but is an indicator of that disease. So if you had some indicator, it could be as straightforward as a gene mutation that you can actually create an assay for and get that information, sequence information, and then you can actually look at a larger patient population, do that assay on that population, and decrease the size of the population that might respond to the drug. So that biomarker is a way of enriching the patient population for responders to the drug. And if you can do that effectively, you can do smaller clinical studies, and you can generate more statistical power with those smaller studies. And then that reduces cost and also accelerates your ability to develop these therapeutics clinically. One of the things that we're working on at Just-Evotec Biologics, and others are as well, but we're we're looking at ways of accelerating from discovery to that proof of concept study. So we want to get there really fast, and we want to get there at relatively low cost. And that's important because we want to do more experiments. We want to do more experiments, or we want to do experiments for lower cost. And we're using machine learning tools in AI to do some of this work as well. And I'm going to talk about some of that a bit later too. But if we can do this really rapidly, and we can do it for a reasonable amount of money, a lower amount of money than it costs us now to do this experiment in people, then we can actually feed back the information that we learn at the clinical stage, that initial clinical stage, and we can feed it back into the discovery process and actually train it or learn from it, adjust our models so that we're actually better at predicting going forward. So we can learn from both our successes and our failures through this feedback learning loop that we can create. Another way, kind of a final way I'll address here about reducing costs, is an opportunity that we're looking at Just-Evotec Biologics in the way that we are pioneering approaches to manufacturing. And so what we're doing is that we're actually designing plants that are smaller, much more efficient, much more flexible, that can be built more rapidly. And if we can do that, then we can delay the investment in commercial manufacturing to a later stage. So that's important because we want as much clinical data to show what the dosing is, or what the demand is going to be. And of course, we want to be absolutely sure that it's going to be an effective drug before we make that investment. So by being able to do the commercial manufacturing capacity to build that, put that in place rather quickly, we can delay again our decision to do that, to make that investment until we have that really important clinical data. So now let's transition a bit and go to what are biologics specifically and how are they used to treat cancer. Kind of a broad working definition of biologics is that a biologic is a pharmaceutical drug product manufactured in, expressed from, or semi-synthesized from biological sources. And there's some major classes I've illustrated here. Actually, it's broader than this, but it gives you an idea. And the vaccines fall in that space, gene therapies, recombinant proteins, cell therapies. You've probably heard about a lot of these. Certainly vaccines we've heard a lot about recently in the SARS-CoV-2 area. There are actually vaccines that are being developed now. I'm quite hopeful that's going to happen soon. I think all of us hope that's going to happen very soon. There are also some recombinant proteins that are antibodies. There are actually antibodies, natural antibodies, that are being fished out of patients that have been exposed to that virus, and then recombinant versions of those are being made being made into drugs. So gene therapies, I'm not going to talk about those, or cell therapies. There's a really should be a great talk around cell therapies by a real pioneer in that area at this conference. So make sure you tune into that talk. I'm going to focus here though on recombinant proteins. It's where I've spent most of my career. It's where a lot of these therapeutics that exist right now have come from. Some of the leading therapeutics, I mean, there's a lot of small molecule drugs that are out there, and chemotherapies and so forth that are out there. But biologics are a whole class that came in and actually effectively treated oncology in different ways, different mechanisms, and quite effective with low toxicity. So very specific technologies, and I'll talk more about why that's the case as I go forward here, but I'm going to focus here. Rituxan, Herceptin, Avastin, Neulasta, Keytruda, these are all leading biologics that are oncology therapeutics, and they have different mechanisms of action that I won't go through here, but they treat different types of cancers, but quite effective. Actually, these are quite effective treatments. Now, antibodies are large molecules. I'm using antibodies as an example, but they're quite a bit more complex than small molecules. In fact, they're vastly more complex because they're really a string of small molecules kind of bound together and then folded in a three-dimensional shape. That's kind of what they are. They're made up of amino acids. They could be strings of 100 to thousands of amino acids that are actually bound together through these things called peptide bonds to make these long chains. And then these chains again are folded, and they have to be folded properly for the molecule to have activity, right? So it's very important, and they're so complex that we can't make them synthetically like we do small molecules. Small molecules can be made through a series of chemical reactions, and because they're relatively simple, you know exactly what you make, you measure it, and it's done, I wouldn't say in an absolutely straightforward way, but it can certainly be done synthetically. And you can't do that with large molecules, especially molecules like antibodies, because they're just way too complex to do that. So we've learned to harness actually cells and the cell machinery over the years to actually have these cells make these drugs for us. So we put the DNA that codes for the drug, the antibody or another protein, into a cell. We get that into the genome of the cell. And then we have ways of actually amplifying the number of gene copies. And then we have ways of actually making more drug per gene copy through some other tricks that we can play that we've actually learned from viruses and how they replicate themselves. But the cell actually has all the machinery it needs to make these long chains of amino acids. It has specific compartments that are inside the cell where these chains fold in the proper three-dimensional shape, and then they are actually modified. Sugars are put on these proteins in specific places, and that's important for their activity too. And then they're actually secreted outside the cell, where we actually purify them, and they will eventually formulate these things. So like a precision tool, these molecules are designed for very specific functions. And so they have to be made in a very precise way. And again, that's when we use cells to do this for us. And they're able to make a phenomenal number of these cells. So we engineer these cells, we engineer them to make literally a few thousand molecules per second of these drugs. So, and then you couple that to the number of cells that we actually culture when we go to these production and manufacturing systems. We're culturing literally 100 or 200 trillion cells in one culture, right? Making a few thousand copies of this drug that we want to make per second. So that's how we get the productivities. We've come a long way, I can tell you, over the past 30 years in our ability to do this. But each of these molecules have to be made precisely. It has to be made, they have to be virtually identical to others, because if they're not, then it changes the activity of the molecule. So like for an antibody, you have binding sites. There are two binding sites. We call it a homodimer. There's two chains that come together, and you have these two binding sites that will bind to a specific therapeutic target. Now that target could be it could be a bacteria, it could be a virus, it could be a cancer cell. But the binding to that target is very precise. So the structure, not only in sequence, but also in three-dimensional shape, and the form that it takes has to be just right. Has to be just right. The other end of the molecule is also quite important. It's called the Fc domain. And that end of the molecule can recruit the immune system to actually work to neutralize or to attack the target. So if it's a bacteria or a virus, it could bring in a macrophage or a cell in the immune system to actually come in and actually digest and get rid of that foreign invader. We're moving into an age now where some of the leading cancer therapeutics and other therapeutics that are biologics are coming off of patent. So Companies that are the innovator companies will put together intellectual property that called composition of matter patents around the drug itself, and also some other patents around formulation and things like that. But those patents have a certain lifespan, and they actually, when those patents expire, it creates an opportunity for other companies to come in and make biosimilars, and it's actually quite a challenging process actually to make a biosimilar, because that biosimilar needs to be remarkably similar to the novel protein. Won't be exact, but very, very similar, both in terms of activity, but certainly in terms of safety. So the good thing about that is that you have a number of companies coming in and making biosimilars, and this competition will actually help drive down the cost over time as well. So what are we doing specifically at Just-Evotec Biologics to make biologics more accessible to a global population? Well, I'm going to talk about our platform. This is a platform for making biologics, and it spans from discovery all the way through commercial manufacturing. So it's kind of the A to Z of choosing the right molecule and then actually manufacturing that molecule to put into people. So we begin actually with the DNA, of course, and we learn from the DNA that's actually in the public domain, and we put this information into computational models that tell us what is the range of what a normal antibody looks like. We use that information to guide us in some of the things that we do in terms of making like libraries that can be screened against a therapeutic target, so we can make billions of molecules that actually can be screened for activity against a therapeutic target. But we have to do this with knowledge. What should these look like? Well, we're actually guided by the hundreds of thousands of sequences that are out there, and we're creating some of those ourselves. We create structural models in computers as well to understand, what does a molecule look like, and is it going to be well behaved? We may want to change what the molecule actually looks like, not its activity, but we may want to change what it looks like so that it will be better behaved from a manufacturing and development point of view. So it may be easier to manufacture, easier to formulate, and we can do that by looking at the sequence that may be a candidate for going into the clinic, and predicting molecules through these computational models that might be superior. We end up making those variants or those different molecules physically. We make them, and then we screen them through some assays, and we choose the best one to move forward. And then we develop processes around all of this to develop these things efficiently, to manufacture them efficiently through very efficient processes that we put in place, expression, production in the upstream, downstream, and so forth. And then transfer those eventually to manufacturing scale. And all of this is done and driven through a common data set that we collect along the way. And this is really the key point, that we collect this information in a common data set through this whole series of steps from discovery through the manufacturing step, and we continue to continually learn from this data set through machine learning algorithms and AI approaches. That helps accelerate this whole process. It puts more of the experiments on a computer and fewer experiments in the lab. They take time and cost resources, people's time actually to do this. So we're trying to do as much computationally as we possibly can in this exercise. The other important point before I leave this slide is that we're trying to learn as much as possible from the DNA, from the most abundant data, like from hundreds of thousands of DNA sequences. And we're trying to get prediction about the methods that we need to put in place to manufacture and develop the molecule. And if you think about DNA, it's this amazing place, amazing library of information, right? And the DNA in a single cell that we have can code for everything that we are as a person. So that gene that codes for the drug contains all the information that we need to make the drug. And our challenge is to extract that information out and to use that information to accelerate the development of the methods that we use to actually do the manufacturing, the formulation, and all the work that we do downstream of that. So I'm going to start really at discovery in an approach that we're using that's actually quite innovative. I don't know of any other company out there that's actually doing this in this way. And we're using a technology called generative adversarial networks. These are GANs. These are actually neural networks that are machine algorithms. So all you data scientists out there, you're going to geek out probably over this, but I'm going to illustrate it by talking about faces. There's actually a website that's called This Person Does Not Exist. So go to the website if you have interest. What this website is showing is that you can have a discriminator, part of this GAN, that you can train on faces, human faces, actual human faces. And then you can have a generator that's part of this GAN that's randomly generating things that are like faces, right? And every now and then it fools the discriminator and gets one through. But then you say, well, that sort of looks like a human face. I'm going to train the discriminator with some more human faces, a bigger set of human faces. And it makes it harder for the generator to fool the discriminator because it's actually more educated. It's actually more educated now. So the generator works really hard, and occasionally it does fool the discriminator, even the more educated discriminator. And so the generator learns from this, right? So it starts to generate more faces that look more human-like. And the discriminator is trained on a bigger subset of human faces. So it's getting smarter. So you've got this sort of cycle going in place, or this juxtaposition of these two neural networks that are kind of battling each other to get better and better and better at creating human faces, right? So I'll ask at this point, which one of these faces is actually a real human face, and which one, and pick it out from the ones that are actually computer generated or through these GANs. So I wish we were this was in person, so I could actually get a show of hands or whatever. But the answer to the question is that none of them are human. These are all generated through this GAN technology, right? So eventually, this whole GAN approach will fool us. We are literally, as people, trained on millions of faces that we've seen over the years, right? But the GAN will get so good that it will actually fool us. That these people are real, but these aren't real people at all. These people have never really existed. Well, we can do the same thing and apply the same kind of principle to molecules. So we can train with this vast number of sequences that are out there in the public domain. We can train the discriminator, make it really smart, and then the generator has to generate molecules that are going to fool the discriminator. And then we can further train, and we can kind of go back and forth, and over time develop a very high quality library that isn't actually human antibody sequence. It's actually computer generated antibody sequence, right? So these sequences have been generated from the GAN, and we don't call them human antibodies, we call them humanoid antibodies. And we can literally the generator can generate billions and billions of different sequences. So we get a lot of variety in these antibodies and what they can bind to and what they can't bind to. And so that creates an opportunity for us to screen this to screen them against any number of therapeutic targets that are out there, because we have literally billions of different sequences, billions of different binding characteristics that some will bind, and then we can fish those out, and then we can look with further analysis to see whether or not that could be a real therapeutic. But this is the direction that we're going. And the really cool part, I don't have time to go into here, is that we can bias these libraries towards specific characteristics, physical characteristics of these antibodies that may confer better therapeutic activity. And that's an area that's a totally new discovery area that we're currently engaging in, but quite excited about. So we also have other computational tools where we can actually look at existing antibodies that may come out of a human being or an animal. We can say, okay, that antibody, it has therapeutic potential, but it needs to be improved. It may need to be humanized, and we can do this with this Abacus tool set, or it actually may need to be improved from a biophysical perspective to be better in terms of our ability to manufacture it and our ability to formulate that drug. So we can actually improve these therapeutics as well, improve them for expression, purification, formulation, and long-term stability. So once we have the DNA sequence that we want, then again we can put this into cells, as I talked about earlier. Really soup these cells up to make thousands of copies of the drug per cell. We can use robotics to develop very efficient assays for purifying that drug and also formulating that drug. Then we can put all of this into small scale down systems that mimic the manufacturing process itself, and then eventually put this into the full-blown manufacturing process, which is really highly productive. So the bioreactor on the left-hand side at the bottom down there, again, could have 100 trillion to 200 trillion cells in that bioreactor, making drug continuously for many days. And then we can continuously purify that drug from the bioreactor through the steps that you see on that move to the right. So we have a highly productive but very intensified process for making these therapeutics, and it's different than it used to be in the past, and I'll talk about that in a minute. But much more efficient, much smaller, in a much better way of actually doing this work. It's not simple. This process is actually quite complicated, but it's an area of focus where we're making very, and the industry is as well, making substantial progress in actually improving the productivity at this smaller scale. And that's important because we want to build smaller facilities, and we want to do it in a much more cost-effective way. So the equipment that we need to use, because we've intensified these processes so much, it's quite a bit smaller than equipment and the big tanks and things that we used to use. And we can fit this equipment into relatively small rooms that we call pods, and they're really clean rooms. And we can reconfigure those clean rooms. We can even, these things are on air bearings, we can even move these clean rooms around. And if we want more manufacturing capacity, we just clone the pod. So we don't scale up into these massive tanks and massive facilities. We just scale out. And that allows us to more reproducibly make more product, because when you invariably scale up, you have all kinds of risk associated with the physical properties of running a process at a smaller scale, and then trying to run that process at a much, much larger scale. So we're completely eliminating that risk. So this is a picture that contrasts the two types of facilities. And facility on the left, you can't see that much of it, but it's more of a conventional manufacturing plant. They don't all look like this, but it's the same kind of principle. These are hardwired, they're hard plumbed. You see the top at the bottom of those photos, those are the top of tanks that actually are multi-story tanks where you have cells that are being cultured. And those things can be 15, 20, 25,000 liters, these vessels, and they're all kind of built in a very rigid and flexible way into these facilities. And so it's an engineering marvel, but it's incredibly complex. And so what we're doing around the facilities is making these things much less complex. We use single-use technology. We don't need central utilities. We don't need all the hard plumbing. We have intensified these processes so that we can operate them at, again, scales where we can put them inside of these pods. We can clone the pods, you know, if we need more. And the key factor here is that we can build the facilities that these pods are in pretty quickly and pretty efficiently, right? So we're literally building a warehouse, and we're doing this now in Redmond, Washington. That is the shell of the manufacturing facility. It's vastly different than commercial manufacturing facilities. In the past, these were billion-dollar facilities. This facility is a fraction of that cost, maybe 10 to 20% of the cost when it's fully built out. And we can do it pretty fast, 12 to 18 months compared to at least 3 to 4 years to build a conventional facility. So we'll build this warehouse out, you know, it'll look something like this in the manufacturing area of the facility. And you can see these boxes. These are the pods that we have these unit operations or this equipment inside of these pods. We have people in there, but not that many people because a lot of this is really automated. So these processes are connected from beginning to end, and we have a high degree of automation that reduces the number of people, also reduces the error rate that we would see normally in a manufacturing setting. So the manufacturing ballroom would look something like this, and we can wheel up tanks that have buffers for purification, or wheel up tanks that have media that we can feed the cells for culturing them and plug them into the pod. So we're really turning upside down in many ways the way that we do commercial manufacturing. Because we're doing all of this and we can do it quickly, again, we can make decisions on investment much later. And those investments, once we make them, are going to be quite a bit less to meet the capacity demands for the drug in the future. So you can kind of sum it up like this. The conventional facilities are quite complex and rigid, the facility design. The process designs are fairly simple and discrete, but they're done at rather large scales, you know, 15, 20, 25,000 liters. Future manufacturing, the facility itself, again, fairly simple and flexible, as I've shown you. The process design, though, is complex and it's connected. But again, we've made huge progress in that space, and I think we can continue to make progress in that space because it's something we can get our arms around, right? We can reduce costs, we can enhance efficiency at the place of the process and not the place of the plant. Okay, so they're relatively small. It's kind of like a mainframe versus a smartphone, right? The smartphone is so flexible and it's so powerful now, getting more powerful all the time, right? In a very, very small package that's easy to use and has a huge amount of utility associated with it. So that's what we're doing actually in the manufacture of biologics. So in conclusion, you know, the reasons for the high cost of biologics, it's quite a complex story, but the single most significant factor is the cost of failure at proof of concept. And I outlined some ways that we're addressing that are in the second bullet. Number of ways, 4 different ways. Biosimilars will also contribute to the reduction of costs, and so will actually the efficiency of the manufacturing process itself. Especially when we get to biosimilar manufacturing, because the cost of biosimilars, there's a huge amount of the fraction of the cost of the biosimilar will be in the manufacturing cost itself. So having a very low manufacturing cost will increase global access through these biosimilar molecules. So just to conclude, there's a lot left to be done, and I would ask that if any of you out there have interest in this, continue to ask questions, to continue to explore this space. There's a lot of work that can be done in the future to make improvements. And it's a very important area to spend a career where you actually have the opportunity to make an impact on the access of life-giving, life-saving therapeutics out there in the future. So if you have an interest, engage, get your education, continue to learn, because the industry and this whole area needs you. So thanks so much. I appreciate the time today and just the opportunity to come and talk to you. I would wish it was in person, but this is, I guess, the next best thing. So thanks so much.
Hi, Ian.
Hi, Lisa. How's it going?
It's going quite well. Thanks for joining me today. I want to remind our audience that you can submit a question about Dr. Thomas's talk, or actually about any of the talks you've heard so far, by going to pollev.com, pollev.com/Nobel56. And you can also participate by text at 22333 and text to Nobel56. About partway through our conversation, Ian will cut away for a question, a poll question that the audience, you can participate in. But first, I want to know something very important. Do you work in this lab?
This lab, I actually do have one section with my students. This is for my Monday afternoon introductory chemistry course. So this is very familiar territory for me.
So tell me, and tell our audience who is unfamiliar as I am with the renovation, is this part of the old Nobel, or is this part of the brand new Nobel?
This is part of the brand new Nobel, and this is one of our kind of designed labs that took a lot of input from chemistry professors, as well as those who actually do run these labs. So, I mean, it's really tailored to the way we teach here at Gustavus. And so we have individualized neighborhoods here for students. So everyone can work at their bench safely. And it's just been really wonderful also to work in a space like this during COVID as well, during kind of more social distance laboratory experiences. So it's been truly a pleasure.
It's kind of an amazing opportunity to test out the flexibility and efficacy of the space. Gee, thanks, pandemic. You keep giving us and giving us. So I wanted to observe that there are a couple of science minutes on the Learning More About Cancer page that if people are interested in just kind of reviewing some of the points that Dr. Thomas was going over, there's one that talks about the stages of drug development, and then there's also one that explains why these large molecule drugs are so much more complicated. And that makes me want to start with my first question, which is, why, tell me how much bigger these large molecules are than the so-called small molecules of chemo. I asked Dr. Thomas this question, and he basically said, they're bigger, and that's why we call them large molecule drugs, Lisa.
That's a wonderful question. And
thank you.
The approaches we can take here as those of us who synthesize these sort of molecules, it's a very different challenge. When we talk about small molecules, these are going to be the sort of things that you find in pills off the shelf, aspirin. They need to be small so they can get through cell membranes and they can get through the targets that they need to get to. And these can be kind of put together through methods that have really, through extensive research over many years, have been kind of honed by organic chemists. Now, in contrast, these biologics, these larger molecules, these are mostly protein-based. And these have essentially is kind of the opposite, where these small molecules are pretty complex. They have a lot of little things that, different considerations. You have to worry about configurations. When it comes to proteins, these are very large molecules, but the backbone and the chemistry between that links these amino acids together is actually the same. But the problem is that there's the human genome is coded for 20 different amino acids. So there's 20 choices at each spot along the way. And so you're combinatorially, if you just have even 5 of these in a row and you have all the 20 choices available, there's 3.2 million ways that this can go. And so from a synthetic standpoint, we would have to build this piece by piece, put one on, stop, rinse it off, put another one on, stop, rinse it off. And that's just not practical from a synthetic standpoint when you need this to be a commodity chemical. And so from a synthetic standpoint, using this recombinant DNA technology that Dr. Thomas kind of was right there when it was starting to kind of come into play when he was at his postdoc at MIT, this being able to inject a gene into a cell and basically turn it into a small chemical factory for you. You're able to take this normal growth media, basically cell food, and turn it into this life-saving drug. It's really, really impressive. Even just kind of that baseline technology that's used in biologics today.
That's astonishing to hear you describe it. Yes, I'm just pondering the process that it would have taken to do that by hand, so to speak. I wonder if this might be a good place to put in our poll everywhere question. So audience, if you go to pollev.com/Nobel56, you can find a poll question there that asks you about what you think about the cost of biologic therapeutics, which is something Dr. Thomas talked about a lot in his talk. And we'll wait for those results to come up, but I want to ask you some more questions, Ian. Say a little bit more, if you can, about, he described machine learning, and it was really interesting. He used the example about faces. But can you talk about that a little bit more, Ian? How have you seen that being used, for instance, in this kind of work?
So that's another important point here. This machine learning, it's starting to make its way into a lot of scientific fields. It's relatively new, and it's really just starting to get its legs in the research community. And I think a lot of industries are starting to kind of realize how powerful it is. So essentially, and what he described as machine learning is that you have essentially these competing processes in order to kind of train this program. And what this program, does is it's trying to take all this information, all these trials, all this training sessions you're giving it, in order to kind of get a right answer, so to speak, depending on the input you give it. So in this case, they're inputting sequences of amino acids, and these proteins are just that, a sequence of amino acids.
Okay.
But that's really the way these drugs work is not really in the sequence, it's how it all bundles together, and the sequence defines that. And so you can't really, that's a very difficult thing to model. If they give you a string of these amino acids and tell you what shape is it going to be, that's a very difficult question. And so this is where this machine learning is coming in and trying to predict, if we put in this sequence of amino acids, what sort of shape of a protein on the back end are we expecting to see? And so if we can train that enough and get it to be pretty accurate, this is going to really cut down that failure rate, which is Dr. Thomas is talking about. It's going to be one of the number one ways to cut costs for the drug development process, in particular for these biologics.
Indeed. So much expense talking about that in that discovery to proof of concept stage. So really just those very early stages. As you heard him describing that, was it surprising to you at all, or did you sort of think, well, that's exactly what I would expect given the difficulty of simply identifying these molecules?
I think just a priori, just thinking about it, doesn't necessarily make sense that it'd be so difficult, because you know your target, you have the thing you're aiming for. You want to design the drug to do this one particular thing. And so you think, well, maybe I can just, look at this, figure what I need in order to maybe shut this protein down that's causing problems in the body, and just go in the lab, make it, and it would work. However, what it turns out to be is that that's a very complicated process, and we can't just look at our target and know exactly what it's going to take to make it, to knock it out, or to do the change we want to see with our biologic drug. And this is where that expense comes in, and it can be a shot in the dark sometimes, but that's not really actually true. A lot of times, this is based on natural products. Is kind of traditionally where small molecule pharmaceuticals comes into play, where you find some sort of product in nature that has some sort of biological activity. Let's say it eases pain. Then you have natural product chemists who will look at this plant and isolate the compounds and try to identify the one compound that eases that pain. And then you get your synthetic organic chemists to come in, make that molecule, and then it's going to be something that you can now produce this on an industrial scale and make a drug out of it. But a lot of times, we try to improve on this, especially when it comes to patents. It gets pretty complicated because, you make a small modification, you can put a patent on it, and if it works better, now all of a sudden this becomes an avenue for you to make money. But making that one small change, try to find something that's a little bit better. That takes a lot of tries. You need to make a lot of molecules, or in this case, a lot of biologics. And this is where that high failure rate comes in, is you just don't know what's going to be the right answer. And so you try a lot of things and kind of hope for the best. This is where you can get those thousands, up to 10,000 candidate molecules. That you need to check that it works, check that it binds, and check that it actually does something even before, and it's safe even before it gets really far into clinical trials.
And of course, when you're talking about life-saving drugs, that time and expense feel very painful, actually, to think about. Well, it looks like we've had some interesting results on our poll. It's amazing to watch how quickly they change when the numbers of contributors are small, but then it's sort of settled out. So I think, Ian, that you can see these results yourself also, can't you? So does it surprise you?
I think the results of this poll, I think there's a lot of cause to be optimistic here. And I think these challenges are really large challenges. Some of these things that Dr. Thomas talked about, these are not small tasks, but the thing about them is that they're not insurmountable. We have technology today that can start working on tackling these problems. We can start training these machine learning programs. We can start working on optimizing the processes that go into making these biologics. And he's already broken ground in Seattle with that new manufacturing facility. I think there's some very real steps being taken here, and I think there is cause to be hopeful. I mean, it doesn't mean that there's not a lot of work to be put in, and it's not going to take a lot of hours of a lot of different types of specialists to do this. But that's exactly the kind of career that Jim Thomas has built, is managing teams like this in order to bring these sort of ideas to fruition. So I think being hopeful I don't think it's unrealistic.
And it was quite moving, I guess I will say, to hear Dr. Thomas talk about his optimism. It feels like, well, he ought to know. And if he's feeling optimistic, then I think I'm all about optimism right now, I guess I have to say.
I think we could use as much as we can.
Exactly. Here's a question maybe out of left field, but I have a feeling you'll have things to say about it. What opportunities do you see for students who are interested in pursuing the kind of work that Dr. Thomas and his company are doing? This is something that he really emphasized in his talk, and I know we have lots of high school and college students in the audience here. So what should they be doing to get in a position to do this work?
I think the reason why Dr. Thomas is outreaching is because, a lot of these the cure for cancer, bringing these biologics to market, looking at these sort of systems that need to take place and need to kind of come into place in order to make this affordable everywhere. This is a really big, multifaceted problem that comes in from many different directions. And so, I think he's reaching out in order to really kind of say, that this is a field that needs all the help it can get. It needs all hands on deck. And it is not inaccessible. This is something where if this is something that you're truly passionate about, put effort in, look into things, figure out what you need to do, look into stuff, get excited, and try to, build a career out of this. And this is something that takes passion, and it takes, a lot of love and joy, because there's going to be times that's going to be frustrating. And there's going to be times where, this is, as Dr. Thomas mentioned, there's a lot of failure that is wrapped up in all this. But after all those failures, you get that success. You can eventually break through and get maybe a new drug to market. And it might be, and if you keep working on these problems, it may be beyond, say, our lifetime. But I think it's really important that students of the next generation kind of take up the mantle and keep making progress.
What are the things they should be studying right now? What are the high school subjects or the subjects at Gustavus or another college that are the most important to be able to contribute?
This is getting into some personal ideas, and maybe it's painted a little bit by the institution that we belong to. But there's a little bit of a multifaceted approach in order to thinking about these sort of problems. Well, a specialist it takes a lot to become a specialist, and really these are teams of specialists that are coming together to make these amazing things happen. But you need to be able to communicate. You need to be able to talk to other people. And so if this is, science courses, you need a foundation there. But you also need problem solving. You need to be able to look at evidence, to look at data. You need to be able to formulate arguments and kind of take the information you get and make an appropriate conclusion out of it. And so there's room for humanities. There's room for writing. There's room for branching out and taking things that you may be interested in. And if you keep this passion for learning alive, you're going to be able to keep going even beyond school. We're lifelong learners, every one of us here, and if we keep engaged in it, there's definitely a lot of progress we can kind of keep making. I of course can state my bias. I took a lot of chemistry. I love chemistry. I teach chemistry. So I would say taking chemistry is a lovely thing. But there's a lot of other specialists here that come into play. And so you can take the courses you need now, but stay engaged, keep reading, and kind of keep on top of it. And you're going to see your passion kind of take you some very interesting places.
Mm-hmm. So something, actually, Ian, I wonder if you have a perspective on is, as I interviewed folks this summer for the podcast, about which I'll say something later, I was struck by the fact that we had folks who had gone the medical route. They were doctors, physicians, and then there were folks who had gone the PhD route, and they were all engaged in drug discovery and development. And I'm just wondering, is there a reason somebody would choose one of those routes rather than the other if they knew that what they wanted to do was be involved in drug research?
I think that's a very good question. And, if it's, I think it's more of a matter of passion and where life takes you, and I think my advocacy is always to find a passion and go for it and take whatever path you feel is right for you. And if you're going through that medical doctor route, maybe some instances, this might not be all, where they thought they wanted to be a practicing clinician, wanted to be one who's, right there in the doctor's office working on patients all the time. And maybe then realized that, oh, I actually really like this drug development and maybe coming up with a new procedure or really working on advancing medical technology, as opposed to someone who might go into a biochemist or really like working with biologics or working in the recombinant DNA field. and maybe kind of come to realize that working in these biologics, developing new biologics might be the way to go. So in terms of people taking their own unique paths and maybe converging somewhere later on, I guess the idea is it's more about the destination, where if you end up there, that's where you end up, and everyone has their own unique path of getting there. And I think those paths inform us and really let us bring a lot of unique perspectives to the table.
Well, thanks very much for your time, Ian. And now we are going to execute one of those moves that makes me very grateful that we have Heroic Productions working with us, and that I'm not trying to now suddenly execute this move. We're going to bring 8 people onto this screen, and then we are going to give just a moment for a new person to appear where Ian has been. And while we're waiting for that last little bit of assembly to happen, I just wanted to tell you briefly about the fact that all these folks who you'll see before you on screen have very graciously this summer spent some time